Okay, guys, welcome to part B of lecture 7. So we are still looking at blood physiology in particular. Today we are discussing immunity. So immunology related to blood. So you know to say this is a function of white blood cells in general. So now, this part of the video, this is part B. So I'll be discussing adaptive or acquired, which is specific cellular mediated immunity. So adaptive or acquired. So like I said, this part of immunology or immunity, there's quite a lot of information. So it will take a bit of time for me to go through all the notes, all the points that I want you to study and to enjoy. There's a lot of mechanisms, so you need to appreciate. So this is very specific. So there's really a lot of information. So just sit back and just be with me as I'll be discussing with you. So I hope you'll be able to understand, because I know you guys are medical students, so I take you to be smart students, so you'll be able to understand. Nevertheless, if you still have a question after our discussion, you can still ask those questions. So we have a lot of platforms that you are able to ask questions. So let's start with adaptive, acquired, or specific cell-mediated immunity. Okay, taking you back, this is the development of white blood cells that are involved in cellular immunity. But this adaptive acquired immunity, I'm interested in lymphocytes in general. Lymphocytes are the ones that are involved in adaptive immunity. Of course, there are also other white blood cells that are involved, but the main point here is the function of lymphocytes. So this diagram I've already explained to you in the first part of this lecture, so I'm not going to repeat myself, but just not to say they're coming from stem cells, then they will differentiate into different types of white blood cells. So these are the cells that are involved in adaptive immunity, the white blood cells. So let's go on. This is just a general picture of adaptive or acquired immunity. So it's just a summary, so we'll go into detail. So I'm just laying a background of adaptive immunity. So you have adaptive defenses. So these adaptive defenses, like I said, when you're talking of adaptive, you need exposure to an antigen. These cells to be activated, they need exposure to an antigen. That's the difference between non-specific and the specific immunity. The non-specific, we said the immunity is there, it's inborn. You don't need any exposure to any protein or any foreign segments or whatever it is, an antigen. You don't need all that. You just have that immunity that is inborn. So it's within your genetic makeup that you produce cells against anything that is foreign. So that is innate. But the adaptive, you need a foreign substance to activate these cells. So there's antigen presentation. So in terms of adaptive defense mechanism in the body, the first thing is antigen presentation that will trigger specific defenses or immune response. So this antigen, after you have antigen presentation, is going to stimulate a specific immune response. So they're very specific. That's why the other name for adaptive acquired, it's also called specific or cellular immunity. So you have two forms of adaptive immunity. You have cell-mediated immunity and antibody-mediated immunity. Cell-mediated immunity, you also have the phagocytes that will present a pathogen or an antigen to the T cells, activating the T cells. Once the T cells are activated, they're going to provide a direct physical and chemical attack to the specific pathogen. So they know to say Once the macrophages or the antigen presenting cells present that antigen to the T cells, the T cells will know the kind of microorganism that is infecting the body. So they will provide a specific immunity against that pathogen. So that immunity is not against any other microorganism, but to be against that specific microorganism that has been presented to them by the antigen presenting cells. So they are very direct. So they are going to provide a direct physical and chemical attack to the microorganism. The activated T cells find the pathogens and attack them through phagocytosis and also release of chemical toxins. So they're going to release toxins against the pathogen. Then they are also involved in phagocytosis. So the activated T cells, they're also involved in phagocytosis. So they know how this microorganism looks like. It will go and attach to these microorganisms. They will release chemicals that will destroy the cells, and they can also phagocytose the microorganisms. That will result into destruction of the antigens that have been presented to them. Then also you have antibody-mediated immunity. This is more of humoral immunity. That is part of adaptive defense mechanism of the body. So the antibody-mediated immunity, there is activated B cells that will give rise to cells that will produce antibodies. So we said the activated B cells will be transformed into plasma cells, and the plasma cells are the ones that will start producing antibodies against the pathogens. So they'll start producing antibodies, and these antibodies, they're going to attack by circulating antibodies. So they are going to attack the pathogen. So they'll be released into circulation. Then they'll be just wandering about wherever they find this specific pathogen, because they are designed specifically for this pathogen. They'll go and attack the pathogen. Okay, so they're very specific and it's very efficient. Okay, so let's set up the background for cell-mediated immunity by looking at the major components of cellular immunity. So the major components of cellular immunity, you have cytokines, major histocompatibility complexes, and other things that will come in. So you have the lymphocytes, the cells that are involved in cell-mediated immunity. And you also have the cytokines, the same cytokines that we have already discussed in innate immunity. So here we won't waste time, but we'll just be pointing out what is the functions of these cytokines with regard to cellular immunity or adaptive immunity. Then you also have certain proteins that are found within the plasma membrane of macrophages and these lymphocytes. So they are called major histocompatibility complex proteins. Major histocompatibility complex proteins. So these are the proteins that are involved in antigen presentation to the lymphocytes for them to be activated. So you have two major classes of major histocompatibility complex. So you have major histocompatibility complex 1 and major histocompatibility complex 2. The major histocompatibility complex 1 is basically found in all nucleated cells. So any cell with a nucleus, they will have major histocompatibility complex 1. So even the normal body cells, the host cells, they can have the major histocompatibility complex 1. But the major histocompatibility complex 2 is only found on antigen presenting cells. So these are special cells that can present antigen to lymphocytes. And also lymphocytes, they can also express these antigen presenting the major histocompatibility complex 2. So you have class 1 and class 2. So we are going to discuss them. You don't need to worry anything. Okay, so where are these lymphocytes coming from? Because we said lymphocytes are the ones that are involved in cell-mediated immunity in general. So they are also coming from the bone marrow. Remember the bone marrow, you have the hemocytoblasts. That is the hematopoietic stem cell that will differentiate into lymphoid stem cell. So the lymphoid stem cell can also undergo mitosis so that they divide into a lot of these lymphoid stem cells. So you can see the lymphoid stem cell here. They can migrate from the bone marrow, Then they will move to the thymus. So as a child is developing, you find that they have a thymus which is functional. In adults, it will atrophy, then becomes dysfunctional, but they're still active in babies that are still developing. So they will start producing a lot of these lymphocytes, especially the T lymphocytes within the thymus. So you can see that some of the lymphoid stem cells, they can migrate via the bloodstream, then they will migrate to the thymus. Within the thymus is very active. You have certain hormones that can stimulate these cells. So you have thymic hormones that can stimulate the lymphoid stem cell that is coming from the bone marrow into the thymus, and then they'll start differentiating into T cells. So you can see the production and differentiation of T cells. Then you have the mature T cells. The mature T cells, some of them will remain within the thymus, just waiting for infection. That's why when there's an infection, the lymph node, they become reactive as well. So you can find these within the thymus and also the lymph nodes. So you can see that the mature T cells from the thymus, they can also be transported by the bloodstream back to the bone marrow, and some of it can be released into the circulation. So you have the peripheral tissues. So these T cells, they can migrate to the peripheral tissues. And then that will trigger cell-mediated immunity. Then you can see that other parts of this cell, the lymphoid stem cells, they will differentiate into B cells and natural killer cells. The B cells are the ones that are involved in production of antibodies. So you have antibody-mediated immunity. That is part of humoral immunity, because you find that these antibodies will be released into the circulation. Then you have the natural killer cells. These are the ones that are killing viral infected host cells and other pathogens that can get into the body. So they are involved in immunological surveillance. So they are released to the circulation. They will just survey pathogens. Whenever they find a pathogen, then they will attack. That's why they are called natural killer cells. So this is where the lymphocytes are coming from. So now let's going into a bit of information. So there are different types of T cells. Like I said, the T cells, they make up about 80% of circulating lymphocytes. So they are the majority of lymphocytes. So we said about 20 to 25% are lymphocytes in terms of differential white blood cell count. So you find that 80% of the lymphocytes are actually T lymphocytes. Remember, you have T lymphocytes, B lymphocytes, natural killer cells. So the 80% of circulating lymphocytes are T lymphocytes. There are different types of T lymphocytes. So you have cytotoxic T lymphocytes, memory T cells, you have helper T cells, suppressor T cells. So these different types of lymphocytes or T lymphocytes, they have different functions. So in general, the cytotoxic T cells, their functions, they attack cells infected by viruses. So they will have a similar function to that of natural killer cells. So they are also attacking cells infected by viruses. So there are certain cells where viruses can enter the cells and they'll start hiding within those cells. The cytotoxic T cells, just like the natural killer cells, they are able to recognize these cells and then they'll attack them. They will stimulate these cells also to undergo apoptosis, so they can also produce interferon gamma and also tumor necrosis factor that can stimulate these cells to undergo apoptosis. So they will destroy viral infected cells. Then they also produce cell-mediated immunity. So they are also involved in cell-mediated immunity. So after presentation of an antigen, so they are going to be activated, then they are involved in cell-mediated immunity. Then we also have memory T cells. So these are T lymphocytes that are memory cells. So the memory T cells, they are formed in response to foreign substance and they remain in the body to give immunity. So they have a memory for that particular pathogen So after activation of the T cells, some of them, they will differentiate into memory T cells. And these memory T cells, they will have a memory of that pathogen to the extent that if next time you have a similar infection caused by the similar antigens, these memory T cells, they'll be activated and then they can easily attack that pathogen. That's why you have these memory T cells, to have the memory of that particular pathogen. That's why I say there are certain diseases that you can suffer from them just once and for all. Why? It's because you have pockets of these memory T cells that will remain within the body to respond to recurrence of a particular pathogen that can gain access into your body again, and then you still have these memory T cells that will attack those pathogens. Then you have T helper cells. T helper cells, you have T helper cell type 1, T helper cell type 2. So in general, the T helper cells, they are going to stimulate function of T cells and B cells. So you find that once they are activated, they can activate the function of other T cells. So they'll produce a lot of cytokines that will have paracrine effect on the other T cells to activate them. Then that way there are a lot of activated T cells that can attack the pathogen. Then we have suppressor T cells. The suppressor T cells, they are more like regulatory T cells. They are going to regulate the function of other T cells. So they're going to inhibit the proliferation, differentiation, and also the functions of the T cells. For instance, after infection, there is a lot of proliferation of these T cells, the activation of T cells, then they'll go to the site of infection. Then you have more specific kind of defense against that pathogen. But after you destroy the pathogen, it's all done. You don't still need a higher concentration or amount of these lymphocytes. So you need to suppress the production of lymphocytes so that it goes back to the normal leukocytes count when you're looking at the leukogram. So how does that happen? There are special types of suppressor T cells, and they are very slow to be activated, so that by the time they're activated, the infection has already been dealt with by the other T cells. So that them now, they are going to suppress these T cells proliferation and activation. So they are going to inhibit function of T cells and B cells. So they are regulatory T cells that control sensitivity of immune response. So they are going to control the sensitivity of the immune response. Otherwise, if this sensitivity is not controlled, it can result into autoimmune diseases whereby your own immune system starts attacking your own cells because you have just produced most of these lymphocytes and you've activated most of these lymphocytes, but there is no pathogen. So what happens? You find that these cells can start attacking your own cells. So you don't want that to happen. So you have the suppressor T cells that will suppress the function of other cells. So like I said, these lymphocytes, they are divided into different types. So you can see this is the same information, but I'm just expanding these cells that are involved in cellular immunity or cellular mediated immune responses. So you have different types of T cells. So let's start with the T cells because we've already discussed them. So I won't waste time here. So you have the T cells. We say that approximately 80% of circulating lymphocytes are classified as T cells. So they are the majority of the lymphocytes. So because they are the majority of the lymphocytes, there are a lot of them. So you have cytotoxic T cells, T helper cells, suppressor T cells, memory T cells, the same cells that we've just discussed in the previous slide. So you can see here, cytotoxic T cells, they attack foreign cells or body cells infected by viruses. So you have a cytotoxic T cell that can attack host cells that are hosting viruses. So they are viral infected cells. So they'll be attacked by cytotoxic T cells. So they have different types of receptors that we're going to discuss, especially when you're talking of cluster of differentiation. Mainly they're going to express CD8 as opposed to CD4 that you are going to find in helper T cells. Okay, so we'll discuss that in particular. So you have the helper T cells. These helper T cells stimulate the activation and the function of both T cells and B cells. Suppressor T cells, like I said, these are regulatory T cells. So they are going to inhibit the activation and also the function of both T cells and B cells. Then we have the memory T cells. The memory T cells are different from the memory B cells. So we have a pocket of memory T cells, then there will be also a pocket of memory B cells after they have been exposed to an antigen. So you have the memory T cells. These are a subset of T cells that respond to a previously encountered antigen. So if you had an antigen now, the next time you have this antigen, they'll quickly respond because they will have a memory of that pathogen or that antigen protein. So they can easily be activated in the attack before it can cause any kind of disease. Then you have B cells. So you also have other subtypes of lymphocytes. So you have the B cells, the natural killer cells. The B cells, B lymphocytes. B cells make up about 10 to 15% of circulating lymphocytes. So after the T lymphocytes, which are about 80, then you have the B lymphocytes, 10 to 15% of the total circulating lymphocytes. Then, like I said, once they are activated, they can differentiate into plasma cells. And these plasma cells, when stimulated, B cells can differentiate into plasma cells, and these plasma cells are the ones that are involved in production of antibodies against the specific pathogen. Remember, adaptive is specific. So these antibodies will be produced specific for certain pathogens. Then we also have natural killer cells, and these natural killer cells, they make up the remaining 5 to 10% of circulating lymphocytes. And you know to say that natural killer cells are involved in attacking viral infected cells and also other pathogens. They are also antigen presenting cells, just like the macrophages, and then also the mast cells and the dendritic cells. All these cells are grouped as antigen presenting cells. Even the B cells is also an antigen presenting cell. So we'll discuss all that. Cytokines, like I said, we discussed cytokines in the part A of this video. So I'm not going to waste much time here because we've already covered that. But just not to say, you have lymphocytes. Once activated, they can also start producing these cytokines with the same functions that we've already discussed. So in general, cytokines are chemical messengers released by tissue cells to coordinate local activity, to act as hormones to affect the whole body. So like we say, the cytokines, they have paracrine effect on the other cells. So they will stimulate other cells to respond. So some of them, they could be interferons gamma that will stimulate the nearby cells to become resistant to the virus so that the virus don't infect them. So some of them, they're involved in chemotaxis to attract other cells to come to the site of infection. Some of them, they can activate the complement system that will attack the pathogens. So all that we've already discussed. So there are 4 functions of cytokines. So to stimulate T cell divisions, to produce memory T cells, accelerate cytotoxic T cells maturation, and also to attract and stimulate macrophages, to attract and stimulate activity of cytotoxic T cells. So all these, you're looking at chemotaxis. They can also promote activation of B cells. So these are just general functions of cytokines. So as we've already discussed this, but maybe just a few minutes on it. So you have alpha interferons, or interferons alpha are produced by cells infected with the virus. So those cells that are infected with the virus, they're going to produce interferon alpha, and they will attract and stimulate natural killer cells and enhance resistance to viral infection. So they're going to attract natural killer cells. And what does the natural killer cells do? They are going to attack these M cells, because they know to say me, I'm infected with a virus. So if I remain quiet, this virus is going to proliferate, then to attack other cells. So before they start attacking other cells, they will send a signal to the natural killer cells to say, me, I'm infected. Can you get rid of me? So the natural killer cells will come in and attack those cells. So they'll produce proteins against the virus. Then they can produce other proteins that will stimulate this cell to undergo apoptosis. The apoptic bodies will be phagocytosed by macrophages and other cells, including the same natural killer cells. So they will attack even the dendritic cells. That's the communication there. Then you also have interferon beta secreted by fibroblastic. So you have the beta interferons that are secreted by fibroblastic, slow inflammation in damaged areas. So if you have damaged cells, they can produce beta interferons. So these interferons, they are also involved in inflammation. So slow inflammation in damaged areas. So when you have damage, you know to say that there will be inflammation. Why? It's because you have the beta interferons, and these beta interferons, they are inflammatory mediators. Then you also have gamma interferons. The gamma interferons, they are secreted by T cells and natural killer cells that will stimulate macrophage activity. So we saw this. I told you to say when there is an infection, the macrophage will produce interleukin 12 that will activate the natural killer cell. The natural killer cell, in turn, it will produce interferon gamma that will go and help the macrophage to attack the microorganism or to get rid of the internalized microorganisms after phagocytosis. Okay, so this slide is just summarizing the cytokines. So you can't really appreciate here. Why? It's because we've already looked at that slide. So I don't have to go through this slide again. So moving on to major histocompatibility complex. This is where it will start getting interesting now, because now you're looking at the actual mechanisms that are involved in cell-mediated immunity. So let's go, guys. Let's look at these things now. The major histocompatibility complex proteins. So like I said, there are two classes of major histocompatibility complexes. So all the cells that are nucleated, they have major histocompatibility complex 1. In the antigen presenting cells, they have another protein which is called major histocompatibility complex protein class 2. So the membrane, these are membrane glycoproteins that bind to antigens. The major histocompatibility complexes, they are proteins. So these proteins, they can bind to antigens. So they are specific to antigens. So they can bind to antigens. They are genetically coded in chromosome number 6. The major histocompatibility complex differs among individuals. So you find that certain people who express certain types of major histocompatibility complex in terms of structure, but they all contain class 1 and class 2, but they can differ from one person to another person. That's why you find that this person can react to this kind of disease in a severe way. Other people, it won't attack them in such a manner. Other people, they are able to get rid of that disease so easily. Others, it will take a bit of time. So there are variation in which these proteins will be expressed. But we all have class 1 and class 2 of major histocompatibility complexes. So you have antigen presentation. So the T cells only recognize antigens that are bound to glycoproteins in the plasma membrane. So these T cells, for them to recognize the antigens, they are not recognizing the antigens from the microorganism itself. No, they need to be activated. So for them to be activated, they can only recognize the antigen that has been expressed by the glycoprotein. And this glycoprotein is the major histocompatibility complex that is found within the cells. So for the T cells to come and attach to the antigen, it needs to be presented by the major histocompatibility complex. that is expressed within different cells. So there are two classes of major histocompatibility proteins. Like I said, there's class 1 that is found in all nucleated cells. Like I've already said, in class 2, that is found in membranes of professional antigen-presenting cells. So you have the professional antigen-presenting cells. So these are the macrophages, natural killer cells, and the B lymphocytes, and the dendritic cells. So these are professional antigen-presenting cells. So they are going to express class 2 major histocompatibility complexes on their plasma membrane on top of class 1. So they can have class 1 and class 2 at the same time. Then this table has so much information that you need to really understand for you to understand cell-mediated immunity. So you have class 1 major histocompatibility complex, then you have class 2 major histocompatibility complex. So let's break it down a bit. So we say that class 1 is displayed by all nucleated cells. The class 2 is only displayed by antigen presenting cells. These are dendritic cells, macrophages, and B cells. So these are special cells which are called antigen presenting cells. They are professional antigen presenting cells. So if a question comes in MCQs, short answer questions to say, list the antigen or the professional antigen presenting cells, don't miss these cells. So you have dendritic cells, macrophages, and B cells. Okay, so we know class 1 on all nucleated cells, class 2, they are only found on antigen presenting cells. That's a fact that we all know by now. So you can see all nucleated cells, they have class 1. Then these special cells, which are called antigen presenting cells, they also have class 2. They express class 2. Then these antigens that are incorporated within these major histocompatibility complexes, they are recognized by which types of cells, which types of immune cells to some extent. So you know to say, those that are expressed by class 1, they will be recognized by naive CD8 cells, the CD8 cytotoxic cells. So the CD8 are the ones that are found within the cytotoxic T cells. So these are special types of proteins that are found on the plasma membrane. It's called cluster of differentiation. So the cytotoxic T cells, they express cluster of differentiation 8. It's a special types of a protein that has got also a function. So you find that on class 1, we say that they are recognized by cytotoxic T cells. That's why even the cells that are infected with the virus, because those cells, they are going to express the cytotoxic, the major histocompatibility complex, class 1, the cytotoxic T cells are able to go and bind to those cells and destroy those cells. Why? It's because it's able to bind to class 1 major histocompatibility complex. Then the antigen that is presented by class 2 major histocompatibility complex can be recognized by naive CD4 cells. So when you're saying naive, it means they are not yet activated. So these are CD4 cells. These are T helper cells. So the T helper cells are the ones that are going to express CD4. So when you're talking of CD4 count, we are just looking at the helper T cells. In the case of HIV, these are the cells that we're interested in because the viruses, the HIV virus will replicate within the CD4 cells. The CD4 cells are the helper T cells. These helper T cells are the ones that can recognize an antigen that is presented by the macrophages for them to be activated. So if the number of CD4 cells or the helper T cells is reducing, it means that your immunity will be compromised as well. Why? It's because you have less cells that can communicate with the antigen presenting cells for them to be activated to start developing immunity that is specific for that antigen that is being presented by the antigen presenting cells. That's why HIV is a very deadly virus. Why? It's because it's going to reduce the number of T helper cells, the CD4 cells. Once they are reducing, your immunity is also reducing, your cellular mediated immunity is reducing, and then you'll be more prone to opportunistic infections because the antigen presenting cells, they won't have any cell to present to for them to activate those T cells. That's the case with HIV. So by now we know to say that the CD8, the cytotoxic T cells, they can only recognize an antigen that is presented by major histocompatibility complex 1 that is found on all nucleated cells. Then the CD4 or the T helper cells, they can only recognize the antigen that is presented by the major histocompatibility complex class 2 that is found on antigen presenting cells. So now when there's this presentation of antigen, the foreign antigen on the major histocompatibility complex. So the one that is found on class 1, what kind of foreign antigen that is found on major histocompatibility complex class 1? So you have endogenous intracellular pathogens or proteins that are made by cancer cells or cancerous cells. So you have the endogenous antigens. It could be a cancer cell. A normal cell has become cancerous because you have factors like carcinogenic factors that can stimulate other cells to become cancerous. So it's a host cell that has mutated and then it has become cancerous. So they'll start growing. So you find that as those cancerous cells are developing, there could be a component of that cell that can be presented to the plasma membrane. So they will send a signal to the cytotoxic T cell to say, I'm part of the body, but the way I'm behaving is not normal. I'm becoming cancerous. Can you destroy me before I destroy the other cells? So the cytotoxic T cells, they are very keen at fighting cancers. Why? It's because they are able to recognize the antigen that is presented by the major histocompatibility class 1. So even the cancerous cells, they will have it. So it will be presented. So you find that this is a foreign protein that will be found on major histocompatibility complex number 1. And also certain segments of pathogens, they can also be presented by these cells. Those infected cells, they'll present to the cytotoxic T cells so that they can trigger them to undergo apoptosis. But the foreign antigen that is found on class 2 that is presented by the macrophages, these are exogenous. Class 1, endogenous, meaning that within. Then those on macrophages or antigen presenting cells are exogenous, meaning that they're coming from outside after phagocytosis. So phagocytosed extracellular pathogens, for instance, from a bacteria. So that bacteria, a component of that bacteria will be presented within the major histocompatibility complex number 2. To present it to CD4 T helper cells to say, the macrophages will be communicating to the T helper cells to say, I'm part of this body, but I've trapped a pathogen which is not part of us. It looks like this. Can you mount an immunity against it? So that would be the communication. So let's go to the foreign antigen on the major histocompatibility complex sends these messages. So the message that this antigen is going to send, what kind of message is it going to send to the cells that are being activated? So the antigen that is found within the major histocompatibility complex number 1, that is expressed by all cells, what kind of information is it going to send to the cytotoxic T cells? Now listen. So we are saying if displayed by antigen presenting cell, then the information is that I belong to self, but I have captured a foreign invader. This is what it looks like. Kill any cell that displays it. So like I said, the antigen presenting cells, they have class 1 and class 2. So on top of class 1, they can also have class 2. So if the antigen is displayed by class 1 on antigen presenting cell, the message there that you will be sending to the cytotoxic T cells is that I belong to self, but I have captured a foreign invader. This invader could be a virus, a bacteria, fungi, parasites, or whatever it is. So I belong to self, but I have captured a foreign invader. This is what it looks like. Kill any cell that displays it. So there are also other host cells, normal cells, that are infected by this virus, that are infected by this bacteria. They are also expressing, they are presenting that antigen on class 1. So the cytotoxic T cells, they have already communicated with the antigen presenting cells to say there is an invader here. So they'll be activated, then they'll start looking for that antigen. So they'll start surveying all the cells of the body. So all those cells that are expressing that antigen on class 1, they'll be attacked by cytotoxic T cells. But if displayed by any other body cell, so this is now any other body cell, what happens? The information that is being communicated is that I belong to self, but I have been invaded or become cancerous. Kill me. So this is the cell that is just surrendering its life for the benefit of other cells that are not infected. Because it knows to say, if its life is maintained, there will be proliferation of that pathogen that will now go and infect other cells. So before that happens, it's going to present that antigen to say, I'm part of self, or the body, but I've been infected by a bacteria, I've been infected by a virus, or I'm about to become cancerous, or I've become cancerous. So before anything goes wrong, can you destroy me? The cytotoxic T cells, they are there now to destroy it. Then if these antigens are being presented by cytotoxic, the histocompatibility complex number 2, what kind of information is given to the cells? The T helper cells now. So this antigen is speaking to the T helper cells to say, I belong to self. The macrophage is telling the T helper cells, because it's the one that is presenting that antigen. So the macrophage is telling the other cells, the T helper cells, to say, I belong to self. Don't destroy me. But I have captured a foreign invader in me. This is what it looks like. Help me mount defense against it. So it's just activating the T helper cells. Once the T helper cells are activated, they'll start producing certain proteins that are specific to that pathogen. At the same time, they'll start producing cytokines to attract other cells, and also other cytokines that will stimulate the more production of white blood cells within the bone marrow. So there will be more proliferation of white blood cells. Then they are also producing certain proteins against that pathogen that is specific because they know how it looks like. So there is communication by these cells. So we proceed. So I hope now this table makes sense as we proceed. So with time, I'll speed up. So let's look at class 1, major histocompatibility protein. So class 1, like I said, pick up small peptides in cell and carry them to the surface. So T cells ignore normal peptides. So if they're just normal peptides, they'll be ignored by the T cells. Abnormal peptides or viral proteins, they're going to activate the T cells, and then that will result into destruction of those cells. So you can see the class 1 major histocompatibility protein here. There's antigen presentation. So antigen presentation by class 1 protein is triggered by viral or bacterial infection of the body cell. Then once you introduce this pathogen into this cell, then there will be the infection results in the appearance of abnormal protein in the cytoplasm. So you have this abnormal protein as a result of infection. The abnormal peptides are incorporated into the class 1 major histocompatibility complex protein as they are synthesized at the endoplasmic reticulum. So once these cells, they sense a foreign protein, they will start producing a lot of class 1 major histocompatibility complex within the rough endoplasmic reticulum. This is where you have ribosomes that are responsible for production of proteins. So some of these proteins are major histocompatibility complex class 1. So they will be now released in form of vesicles. They'll be transported as vesicles to the plasma membrane, and these proteins, they're incorporated within the membrane of the vesicle. So once they are translocated to the plasma membrane, they'll become part of the plasma membrane. So they'll be presented as major histocompatibility complex number 1, but with an antigen attached to it. So you can see here, the antigen was introduced into the rough endoplasmic reticulum, and then it was incorporated within the major histocompatibility complex number 1. Then it will be transported by dynein and kinesin to the plasma membrane, and then it will be incorporated within the plasma membrane. So it will be presented like that. So the cytotoxic T cells will be able to come and recognize this pathogen. Then it will know to say that this cell is infected with bacteria or a virus. Then it will produce other proteins that can attack this cell. That will result into apoptosis, or it will produce lymphocins and other complement proteins that can attack the infected cells to kill them. So you can see here the abnormal peptides are displayed by class 1 major histocompatibility complex protein on the plasma membrane. Then after export to the Golgi apparatus, the major histocompatibility complex reach the plasma membrane by transport. So this is number 4. Then this is the final one where you have recognition of the antigen by cytotoxic T cell, and then it will attack the cell. That's how it works. So we proceed. Then you have class 2 major histocompatibility complex. You have antigenic fragments that are presented by the class 2 major histocompatibility complex. This is found within the antigen presenting cells. So you have from antigenic processing of pathogens by the macrophages or antigen presenting cells, they're going to bind to class 2 proteins, just like class 1, the same process. Then they'll be inserted in the plasma membrane to stimulate the T cells. So the T cells, we're talking of the T helper cells that will be stimulated by the antigen presenting cells. These are macrophages, natural killer cells, plasma cells. Okay, then after antigen presenting cells present the antigen to the T helper cells, then there will be activation of the T helper cells. So these are responsible for activating T cells against foreign cells and proteins. So this kind of immunity is very specific for that particular pathogen. So again, this is just a summary of how the antigen presenting cells are going to present the antigen using the major histocompatibility complex class 2. So let's just look at this diagram, just a bit of it in general. So you have class 1, class 2 major histocompatibility proteins. This is a antigen presenting cell. It could be a macrophage, natural killer cells, or plasma cells or B cells. I mean B cells, not plasma cells. So you have this macrophage or antigen presenting cells that are internalizing the bacteria. So there's phagocytic antigen presenting cells that is going to engulf the extracellular pathogen. So it will be internalized, and you know to say that this vesicle containing the pathogen is going to fuse with a rhizosome, just the normal process, cellular process. And then within the rhizosome, you have proteins that are capable of digesting anything, be it carbohydrates, lipids, nucleic acid, proteins. So they are capable of digesting anything. So they'll be able to digest the pathogen. Then you can see after digestion of the pathogen, this pathogen, part of it is going to fuse with endoplasmic reticulum that is producing class 2 major histocompatibility protein that is enclosed within a vesicle. So it's going to fuse with it. Then this major histocompatibility complex class 2 is going to bind to part of this pathogen. Then it's going to be translocated to the plasma membrane. So it will be part of the plasma membrane presenting that portion of the antigen. So now it's going to present the antigen to T helper cells. So you can see here that the antigenic fragments are bound to class 2 major histocompatibility complex protein. Then it will be translocated to the plasma membrane. The antigenic fragments are displayed by class 2 major histocompatibility proteins on the plasma membrane. So here now there'll be activation of T helper cells because there's that kind of communication. So these are the plasma membrane of T helper cell and cytotoxic T cell. So like I said, they have an extra kind of protein that is also associated with these major histocompatibility complexes. So you can see here in T helper cells, they have CD4. So this CD4 is just a single polypeptide or a single protein that can also bind to other structures from the macrophages, and it's going to facilitate the presentation of the antigen to the T cell receptor. So you can see the T cell receptor here that will bind to the antigen that is presented by the class 1 major histocompatibility complex. I mean class 2, because you're looking at the CD4 T helper cells. So they are only responding to the antigen presenting cells. So this is the class 2 major histocompatibility complex. But the cytotoxic T cells, they will express CD4-8, and CD4-8 is heterodimer, meaning that it's composed of different types of proteins, as opposed to the single protein, which is a CD4. CD8, you have 2 proteins that can also bind to other receptors, but it's also going to facilitate the presentation of the antigen to the T cell receptor. Then there is activation of these cells that will respond like we've already discussed. So we proceed. So just a bit of information on cluster of differentiation markers. So these are CD. The CD are the proteins that are associated with the lymphocytes, especially the cytotoxic lymphocytes and the T helper lymphocytes. So these are the ones that will have cluster of differentiation. So you have different types of this cluster of differentiation, over 70 types. So in T cell membrane, this is where you find cluster of differentiation. So there are more than 70 types designated by an identifying number. So you have CD4, CD8, CD1, CD20, CD30. So there are a lot of them. So the most common one are CD8 that occurs on the surface of cytotoxic T cells that will bind to major histocompatibility complex 1 that is expressed by all the cells. That's why the T cells is able to bind to all different types of cells. Then you have CD4 that occurs on the surface of helper T cells that bind major histocompatibility complex number 2 protein. I think we've already discussed that. So the CD8 and the CD4 proteins, they will facilitate the binding of the major histocompatibility proteins to the T cell receptors, like we've already seen in that diagram. So you have the molecular mechanism of antigen recognition. So they are also playing parts in antigen recognition so that they can be activated. And they also foster lymphocyte development. The activated CD8 cytotoxic T cells, so the activated CD8 cytotoxic T cells kill their targets directly. So once they are activated, they will go and search for those target cells that are infected, then they'll kill those cells, which is different from T helper cells. So the activated CD4 helper T cells, they will produce and secrete cytokines that will activate other lymphocytes. So they will produce cytokines. They can also produce lymphotoxins that will attack these cells. So the two important CD markers, I think it's the same information. This is just extra information for you to read on, but it's just basically the same information as the previous slide. Then these, to some extent, we've already discussed. So you can see the CD4 T cells, they have a receptor, T cell receptor that can bind to the antigen. So if this antigen is presented by antigen presenting cells, you have class 2, major histocompatibility complex. So it will activate the CD4 T cells after that interaction. So you can see here, antigen processing by the antigen presenting cell, and then it's going to express it, and then that will result into activation of T cytotoxic T cells or CD8 T cells. Then the other one here, you can see that this is just an infected target cell. So it has got this bacteria or this virus. So it's going to express that component of the antigen within the major histocompatibility complex class 2. Then it's going to present to CD4 T cells. So the CD4 T cells is going to produce interferon gamma. The interferon gamma is going to destroy the pathogens. At the same time, it can also produce tumor necrosis factor that will result into apoptosis of this cell. The cytotoxic T cells or CD8 T cells, once they are activated, they will produce cytolytic. These are perforins, just like the complement system. They can also produce FAS ligands. If you remember in apoptosis, the death receptor pathway, we had the tumor necrosis factor and the FAS ligands that can go and bind to the receptors or the death receptors that will trigger activation of adapter protein, and that will activate initiator caspases that will initiate apoptosis by activating the executional caspases that will execute apoptosis by going into the nucleus to activate the endonucleases, and the endonucleases, they will digest the DNA and the cytoskeleton, the cell becomes so weak, and then there'll be formation of apoptic bodies. And that apoptic bodies will be phagocytosed by the adjacent cells and also macrophages. So it's also going to trigger apoptosis of this cell. It's also going to produce cytokines like interferon gamma and tumor necrosis factor that is also involved in apoptosis. So you can see interferon gamma is also involved in opsonization of the bacteria, so that it's very easy for this cell now to take care of it or to destroy it after that. Okay, moving on. So we are still looking at cell-mediated immune response. So the general overview of cell-mediated immune response. So the information that we had earlier on was just the general information, but now just going in detail, just to apply that information by looking at cell-mediated immune response. So we have antigen presentation. By now you have an idea how that is taking place. Then you have antigen recognition, you have T cell activation, antigen binding. There's also co-stimulation. Co-stimulation is because you have two receptors. So you can have major histocompatibility complex and the cluster of differentiation that will result into co-stimulation of cytotoxic T cells or T helper cells. Then you have T cell proliferation after activation, of course, because this cell will be activated, so it will start proliferating so that you produce more T cells to combat the infection. Then the T cell differentiation. Okay, so the antigen presentation, this is the first step in immune response, like we've already seen. So extracted antigens are presented to the lymphocytes, like we saw, or attached to the dendritic cell to stimulate lymphocytes. So you know to say these are antigen presenting cells by the macrophages, dendritic cells, or B lymphocytes. Then they can be phagocytic or non-phagocytic antigen presenting cells. So these are antigen presenting cells. So you have the professional antigen presenting cells, which I gave you. There are three here in general: the dendritic cell, the macrophages, and the B cells. These are called professional antigen presenting cells. The antigen presenting cells, they will process and present the antigen peptides. They'll process and present the antigen peptide or a protein to the T cells in association with major histocompatibility complex 1 and major histocompatibility complex 2. So because you also have major histocompatibility complex 1 or major histocompatibility complex 2, then on top of that, you also have the CD, the class of differentiation. So there will be co-stimulation. So thereby it's going to result into co-stimulation of these cells and the activation of these cells. This is what I mean by co-stimulation. Then you have antigen recognition. After that, the T cells, they need to recognize that antigen. So after antigen recognition, the inactive T cell receptors, they will be activated. So they are going to recognize class 1 and class 2. Then they are going to produce certain proteins that are specific. So you have double recognition because of others are being presented by class 1, others are being presented by class 2. Double stimulation or co-stimulation if you want. So binding occurs when major histocompatibility complex protein attaches or matches antigen to the T cell receptor. So they are specific to that antigen. Then they'll be able to be activated. Once there's presentation, you can see the CD4 is also taking part, and also the CD8 when you're talking of cytotoxic T cells. There will be activation of the T helper cells that will result into clone formation or proliferation of these T helper cells. In some of them, they will differentiate into memory T cells. So after activation, like I said, there's activated T cells that are going to enlarge and multiply or proliferate. Then the newly expanded population of T cells, they're going to differentiate into different types of T cells, the cytotoxic T cells, the memory T cells, and also the regulatory T cells. The regulatory T cells are the last ones to be produced because they are going to suppress immunity. You don't want to do that when the infection is still at its peak. So they'll be the last ones to be produced. These are suppressor T cells, and there's also production of helper T cells. Okay, so this diagram is more specific here. So you have antigen presenting cells, the macrophages, the dendritic cells, and B cells again. So an example, you have the dendritic cell that is presenting this antigen on major histocompatibility complex number 2. So you have this major histocompatibility complex number 1 and 2 that is presented by the antigen presenting cells. So you can see here that the major histocompatibility complex 2 or 1 that is being presented by the antigen presenting cell, in this case, the dendritic cell. Then you have the T cell that will be activated. So the T cell, they have a receptor that is specific to that antigen. On top of that, you also have cluster of differentiation 8 or 4, depending on the cell, 8 cytotoxic T cell, 4 T helper cell. So there will be communication. So they will bind and then they will know how the antigen looks like. There will be geo signal. Why? It's because it's being presented by class 1 and class 2 at the same time. And also you can see that the class of differentiation 28 is also participating here, that can bind to other receptors. So that will result into geo signal or stimulation. And this cell will be activated. Depending on the type of the cell, it will start producing interleukins 2. Interleukins 2, it has got autocrine function. So you can see that it will produce interleukins 2. Then interleukin 2 will be released to the interstitial space and then to go and bind to the receptor of that particular cell. So this is an example of autocrine activating this cell. And once this cell is activated, then there is T cell proliferation. So you have clonal expansion and differentiation of these T cells. So you will produce more of CD8 cells and CD4 cells once they have been activated. But you can also see here that there are these cell adhesion molecules that are taking part. So the cell adhesion molecules, they are also specific. They can bind to each other to secure so that there is ample time for this antigen presenting cell to present the antigen. Then there will be a reaction taking place. So you can see here after activation, so you have the infected cell or a tumor cell or a foreign cell with that antigen. So after activation, they have been activated. So you have these cytotoxic T cells that can go and bind to the class 1 because you're talking of a normal cell that is infected by these pathogens. So they will go and bind to class 1, activation of the T cells. So these are cytotoxic T cells because you're talking of just class 1 cells. They are not being presented by the antigen presenting cells. So after activation of the T cells, they'll start producing FAS ligand. So you can see this FAS ligand that can bind to death receptors that will trigger apoptosis. At the same time, this cytotoxic T cell can also produce other proteins like perforins, and these perforins are complement proteins. Perforins will be released. These perforins will go and be inserted within this cell that is infected. It will create a space or a gap, more like a gap junction. Then at the same time, it's producing certain enzymes that will go into that cell to enhance apoptosis. So you can see this cell that is infected now, it will undergo apoptosis, and those apoptic bodies will be phagocytosed by a macrophage. So it's just the same information here. This is just a pathway for CD8. This is a pathway for CD4. So it's basically the same information that will result into activation of macrophages, because once the T helper cells are activated by this, they'll start producing interferon gamma, and the interferon gamma will go and activate more macrophages. So on top of that, they are causing opsonization. So these bacteria, they don't have the capacity to show their virulence because they are coated within this interferon gamma. So they'll be easily internalized by the macrophages and then they'll be destroyed. At the same time, interferon gamma can signal other cells to become resistant to the virus, so they won't be infected. Okay, so this is just the same information that we've already discussed. So we proceed to something new. So you can see the cytotoxic T cells, they'll start producing poisonous lymphotoxins that will destroy the target cell. Then activation of the gene, target cell, that cause cell death. You're talking of activation of the gene that are responsible for autolysis, or not autolysis, but apoptosis of these cells. Then they can also release perforin. In this perforin, they are released to destroy antigenic plasma membrane. So to punch holes, to perforate the plasma membrane to allow movement of other things into the cell and then to be destroyed. So it's basically the same information. So this is what I'm talking about. So you have cytotoxic T cell that binds to infected cells. So they are going to bind to the infected cells because they are being presented by class 1 major histocompatibility complex. Then they will signal the T cells. These T cells will start producing perforins or certain chemicals that will destroy the cell. So the perforins will create holes within the plasma membrane and this cell will be destroyed. Then there will be memory T cells that will be produced again. So once the T cells are proliferating, there is a clone of T cells that are developing. So some of those T cells will develop into memory T cells. So these are produced along with cytotoxic T cells, and then they stay in circulation, immediately form cytotoxic T cells if some antigen appears again. So if there is an antigen that is appearing again, the memory T cells, they will transform into cytotoxic T cells to attack that pathogen before it causes any harm, before it causes a disease. Then you have suppressor T cells. They produce and secrete suppression factors that are going to suppress the immunity or the immune system. So there will be immunosuppression because of the suppression factors that are going to inhibit responses to T and B cells. After they act after initial immune response. So they are going to limit immune reaction to a single stimulus. So you're talking of precision and also control this immunity. So this information I've already shared. So let's proceed. Even here, the destruction of the cells by these cells, I think I've already seen. I've already discussed the function of perforins that are released to destroy the plasma membrane, the cytokines that it is to stimulate apoptosis, and then you also have lymphotoxin that are released to cause this disruption of metabolism. So there's disruption of metabolism. So this cell can't metabolize because these pathogens that are infecting this cell, they are depending on the metabolism of this cell, especially the viruses. So you find that the cytotoxic T cell can produce lymphotoxins that can interfere with the metabolism of the cell so that it will stop producing proteins. Then these pathogens will be more vulnerable to other cells that will destroy them. So this slide we've also discussed to some extent, so we proceed. These are just extra information for class 1 and class 2, which we've already discussed. Okay, so even that, in this, we've already discussed. Let's move to something new. Okay. So like I said, immunity, there is too much information. So you can't just avoid it because we need to know it all. We are training medical doctors, so you need to earn it for you to be called a doctor. You need to know this information. You need to be able to explain it even far much better than myself as your lecturer. So we proceed. So now let's look at still discussing adaptive immunity, but moving on from cell-mediated immunity to more of humoral response of adaptive immunity. So you're talking of the fluids of the body. Plasma, interstitial, intracellular, fluids of the fed spaces in cavities. So what sort of immunity are you talking about here? So here mainly we are looking at the production of antibodies. They're the ones that are involved in humoral response of adaptive immunity, which is a function of B lymphocytes. Once they have been activated, then they will transform or they will differentiate into plasma cells, and the plasma cells will become so big and active with a lot of rapid reticulum, they will result into production of more antibodies. Humoral immunity arises from B cells, like I said. So B cell sensitization is the first step. Responding antigens or corresponding antigen in interstitial fluids bind to B cells receptors. So these antigens, they'll bind to B cell receptors, and B cell prepares for activation. preparation process is called sensitization. So this preparation process is called sensitization of B cells. So during sensitization, antigens are taken into the B cells, they are processed. Then after they are processed, they will reappear on the surface, bound to class 2 major histocompatibility complex. So that's why I said that the major histocompatibility complex, they are also expressed by the lymphocytes themselves. So the B lymphocytes, they also have major histocompatibility complex class 2. So they will internalize the pathogen, because you know to say that the B cells, they're also phagocytic cells, they're also antigen presenting cells. So they will internalize the pathogen, they'll process it, and then they'll also present the antigen on class 2 major histocompatibility complex. So you can see the inactive B cells that can be activated by this antigen binding to antibodies. So on the plasma membrane of these plasma cells, you also have antibodies specific for antigens. But the first one, you can see here that the antigens bound to the antibody molecule, then they'll be processed, they'll be internalized, and then they'll be processed, and then they'll be incorporated into the class 2. So this is now a sensitized B cell, the one that has been sensitized after processing of the antigen. Then they also require the T helper cells, because they are presenting that antigen to the T helper cells. So the sensitized B cells, the sensitized B cell is prepared for activation, but needs the helper T cell. They need, okay, let me just, okay. So we are saying that the sensitized B cells is prepared for activation, but they still need the helper T cell activated by the same antigen. So they're going to present the antigen to the helper T cells to activate the T helper cells. Then in return, the T helper cells can produce certain chemicals to reactivate the plasma cells, or the B cells, for it now to be transformed into plasma cells. So B cell activation. The helper T cell binds the major histocompatibility complex, and then it will start producing cytokines that will promote B cell activation and division. So the plasma cell has internalized the pathogen, processed the pathogen, present it on the major histocompatibility complex class 2 to the T helper cells. The T helper cells, it will go and bind, then it will be activated to start producing cytokines, and these cytokines will have an effect on these B cells. And the B cells will be activated, and then they'll start proliferating and dividing. So you can see here the presentation of the antigen by the B cells to the T cells via the major histocompatibility complex class 2. Then the T cells, once activated, to start producing cytokines. So you have co-stimulation again here. The cytokines will activate the T cells, and then the T cells will be sensitized, and then they'll start dividing. So the activated B cells will divide into plasma cells and memory B cells. So some of the clones will become plasma cells, and some of them they'll become memory T cells, memory B cells now. So these memory B cells are the ones, once that infection comes again, a different time, they will be activated into plasma cells, and then they'll start producing antibodies. Just like we had the memory T cells, once there is reinfection, they can be activated into cytotoxic T cells and attack the same pathogen. Here again, the memory B cells, they can be activated once you have reinfection. They will be activated in the attack those pathogens by producing antibodies against them. So you can see here that the activated B cells will be transformed into plasma cells and start producing a lot of antibodies. The plasma cells synthesize and secrete antibodies into the interstitial fluids. The memory B cells, like the memory T cells, remain in reserve to respond to the next infection, like I've already said. So this is what is happening. So you have the B cells that can, this is a naive B cell that can bind to a bacteria because they have IgD and IgM monomer on the plasma membrane. So they can bind to these pathogens, then they'll internalize the pathogen, Then the pathogen will be destroyed by the lysosomes. Then components of that pathogen will be incorporated within the histocompatibility class 2. That will be expressed within the plasma membrane to activate the T cells. These are T helper cell type 2. So the T helper cells, they have CD4 and a receptor. So this receptor will bind with the antigen. There is co-stimulation or cooperation of the T helper cell and the B cells. So they'll start producing cytokines. Some of the cytokines will activate the B cells, and some of the cytokines will go and stimulate more production of white blood cells within the bone marrow, and some of them, they are cytokines that will attract other cells to come to this site of infection. So you can see here now that the plasma cell, the B lymphocytes that have been activated, then they'll undergo proliferation and differentiation into plasma cells. The plasma cells will start producing antibodies, different types of antibodies, depending on the pathogen. So these antibodies will be specific for this pathogen. So they'll start producing IgM, IgG, IgA, and IgE. So you have IgM, IgG, IgA, and IgE. These are antibodies that are involved in humoral response. That will go in circulation now looking for these pathogen, because they are specific for the pathogen, they are able to attach to them, and they're going to neutralize the infection. Then they are also going to destroy the pathogen. Okay, so I think it's just the same process here. So you have the antigen presenting cell that has got internalized the pathogen. They're going to be expressed on class 2 major histocompatibility complex. Then you have T cell receptor that can bind to the pathogen, and CD4 that is found within T helper cells type 2. So they'll be presented, there'll be activation. The T helper cells will produce cytokines that will activate the B cells that will undergo proliferation and differentiation into a clone of plasma cells. So there'll be a lot of them. They'll start producing antibodies that are specific for this pathogen. So they can go and attach to that pathogen. So they will neutralize the pathogen, and then they're also involved in opsonization of this pathogen. So you can see the secreted antibody molecules, and once this pathogen has been opsonized, it can easily be phagocytosed. But when you expand on the plasma cells, you can see that you have a lot of endoplasmic reticulum, because it's involved in the production of proteins. They have ribosomes on them, and these ribosomes, there will be messenger RNA, transcription in the nucleus, translation into a protein that will become an antigen. This antigen is specific. So this once this translation into a protein, the antigen will be transported to the Golgi apparatus for modification. So it will be transformed into this Y shape of the antigen. So you have different types of antigen: IgA, IgD, IgE, IgM. Okay. And some of the clones, so you have a clone of memory B cells. So these will be memory B cells. So they'll be ready to respond for reinfection. Once there is reinfection, they will quickly be activated into plasma cells, and then they'll start releasing these antibodies that are specific for that reinfection. So it won't go very far once you have this immune system. This is just showing the process of production of antibodies. So you know by now you have the nucleus, you have transcription into messenger RNA to go to the rafting endoplasmic reticulum that contains ribosomes. Then the message that is brought in by the messenger RNA will be translated by the ribosomal. So you have transfer RNA, and that transfer RNA is found at the center of the ribosome, the 40S and the 60S subunit. At the center, you have the transfer RNA that is going to translate the message that is brought in by the messenger RNA into a particular protein. So you have these peptides, and these peptides, there are two peptides that are being produced here. You have the light chain peptide and the heavy chain peptide that will become an antibody. So they'll be transported to the Golgi apparatus for modification. Then you can see here, there is assembly of this light chain and the heavy chain. So you have light chain, heavy chain, light chain. That will be transported to the Golgi apparatus, and then the Golgi apparatus is involved in modification of this protein into a particular shape, then to become an antibody. Then it will be transported in form of vesicles. So you have different types of antibodies that are produced here. For instance, here there's production of IgM or IgA. Then you have these antibodies that will be secreted. The IgM, like the ones that are found, the antibodies that are specific for antigen that is found within the red blood cell, they are also called IgM. So you can see here, the IgM, they are not necessarily bigger than the IgG. Remember, the IgG are able to cause the placenta to go to the fetal circulation. The IgM, they are unable to do that. Why? It's because they precipitate. So you can see that there is a assembly of the IgM. So you can see they form like a star because most of them, they're going to attach to one another. Then because of that, they can't really cross the placenta to go to the fetal circulation. That's why you don't have antibodies that are specific for antigen that are found within the red blood cell in the fetus yet. Why? It's because you can't have passive immunity from the mother to the fetus with this IgM, but you can have passive immunity with reference to the IgE. So you can see the IgE, they are secreted, but they will exist as an individual antibody. They don't cramp to one another. So you find that they are able to cross the placenta to go to the mother. So this is just the process of production of antibodies by the plasma cells. So there are two pairs of polypeptide chains. I think I'm getting tired now. So there are two pairs of polypeptide chains. There's one pair of heavy chain and another pair of light chains, like I said. So each chain contains a constant segment and a variable segment. So a constant segment, it doesn't change for this antibody, but the variable segment is the one that is specific for a certain protein, which is an antigen. So looking at the different types of constant segments, you have different types of antibodies. So you can see a general structure of an antibody. You have the constant segment of the light and the heavy chain. So this is a constant segment of the heavy chain. Then you also have the constant segment of the light chain. And then you have the variable segment that is specific for an antigen. So it has got a free site where it can bind to the antigen. So here an antigen can bind, even here another antigen can bind. Then you can see that the heavy chains are connected by disulfide bonds. So you can see these disulfide bonds that are connecting different portions of the antigen. So this modification takes place within the Golgi apparatus. Remember, the Golgi apparatus can add a moiety, and then it can also result into addition of the sulfide bonds, just like within the raffinose reticulum, we can also have modification whereby there is addition of the sulfide bonds. So I can see this structure of the antibody. Then you also have other sites where other things can bind. For instance, the heavy chain has got a site for complement binding site. So the antibody is able to activate a complement protein. Remember, I told you to say antigen antibody complexes, they can activate a complement protein. So it will have a binding site for a complement. So complement binding site. Then you also have another site where macrophages can bind. So the antibodies can also bind to the macrophages. So as the macrophages are moving, they are moving with these antibodies. The antibodies are able to bind to the pathogen, which is a bacteria or a virus. Then these macrophages will be able to internalize the pathogen because the antibody can bind to the macrophage. It can also bind to the antigen. So that will also enhance the function of macrophages. So it will be very easy for the macrophages to internalize a lot of these antigens or pathogens once the antibodies bind to them. So you have different types of antibodies depending on the structure of the heavy chain. So you have the heavy chain constant segment. So the constant segment is the one that is going to determine the 5 types of antibodies. So you have IgG, IgG, IgE, IgG, IgE, IgD, IgM, and IgA. So when I was a student, how I could remember this, I could just say antibodies. There was a name for it. So we said, okay, how do you remember this? So you can just come up with a word. For instance, you can say GDAM, GDAM, GDAM. So GEDAM, GDAM. So this GDAM, I mean like this. How you pronounce it, it's just up to you. So for me, I could remember that the immunoglobulins, there are 5. So I had this word to remind me. GDAM. GDAM. So IgG, IgE, IgD, IgA, IgM. So GDAM antibodies. There are 5. So you can't miss anything there. Okay. So we proceed. We're almost done now. Okay, so these antibodies are found in body fluids. So once they are produced by plasma cells, they'll be released into the body fluids. That's why they're part of humoral immunity. So they are determined by constant segment, like I said, and then they have no effect on antibody specificity. So the constant segment, they don't have any effect on antibody specificity. So they are not specific for anything, but of course, they have a binding site for complement proteins and also macrophages. You need to know that. Then you have the variable segment of the light and heavy chains. So the variable segment of the light and heavy chains are the ones that are specific for a particular antigen. So these are they determine the specificity of antibody molecules. So how they're going to bind to those antigens. So the binding site of the antibody is determined by the variable segment of the light and the heavy chains. So you have the variable segment, the light and heavy chains. So these are the binding sites. So there are free tips of 2 variable segments. So they form antigen binding sites on antibody molecules, which bind antigenic determinant sites on antigen molecules. So this is where they're going to bind with the antigen. So you have these sites. So you can have a complete antigen, which is different from a partial antigen. Okay? So there is a complete antigen and a partial antigen. A partial antigen, they're also called heptins. So heptins are partial antigens. Then you have a complete antigen. Why? It's because the antibody has got 2 binding sites. So for them to bind to the antigen, they need to bind to 2 sites. So a complete antigen, it has got 2 antigenic determinant sites. So it's able to bind with the antibody. But the partial antigen is just partial. So it's just one part. It's just going to bind to a single binding site on the antibody. But what is going to facilitate this partial antigen to bind to the antibodies? There is this partial antigen must attach to a carrier protein. So there is a carrier protein that is going to attach with the partial antigens or the heptins, then to appear like a complete antigen, then it will be able to bind to the antibodies. So the antibodies, they are produced, the antibodies produced to attack both heptins and carrier proteins. So you need to know that these antibodies that are produced, they will destroy both the partial antigen and the carrier protein. So sometimes the carrier protein could be a normal cell or a normal protein in the body, but the antigen, they are going to attack both the carrier and the heptin. And that can result into antibody that is attacking normal cells. So we have these antibodies that is attacking normal cells. Why? It's because the carriers could be a protein of a normal cell. So it will go and bind with the antigen, the partial antigen, then the antibody will bind to the partial antigen and the carrier protein, which is part of the normal cell. So these antibodies will attack the normal cell. An example is penicillin allergic reaction as a result of these partial antigen and carrier protein. Okay, so I'm just going to summarize that in a diagram. This is what I mean. So you can see this is a complete antigen. So they have two antigenic binding sites to the extent that it can go and bind to the antibody. The antibody is Y-shaped, so it needs both sides to be attached to the antigen for it to destroy that antigen. Then you have the partial antigens, which are called heptin. So the heptin can only bind to one side, but for it to be destroyed, you need another molecule to go and bind to the other side, because the heptin is just binding on one side. There's a carrier molecule, the carrier protein that could be part of the normal cell that will bind to the heptin. Then they will complete the binding to the antibody that will result into destruction of the heptin and also the carrier protein. So if this carrier protein is part of the normal cells, that cell will be also destroyed. So the antibody molecules can bind to heptin, the partial antigen, once it has become a complete antigen by binding with the carrier molecules, like I've already explained. So the functions of antigen antibody complexes, think of a bit fast. So what are the functions of antigen antibody complexes? Once the antibody attaches to the antigen. So what are the functions of antigen antibody complexes? The first one is neutralization of antigen binding site. So they're going to neutralize the antigen, because the antigen, they have this binding site that can go and bind to the cells, then they'll invade the cells. So if the antibodies are binding to those binding sites on antigen, then they are neutralizing the antigen. So neutralization of antigen binding sites. The other function, precipitation and agglutination, formation of immune complex. So you're talking of precipitation. There are other antigens that are dissolved within the plasma or fluids, so they can go and bind to those antigens and then they'll precipitate those antigens. And that can also result into agglutination reaction. Activation of complement, because the antibodies, they also have a binding site for complement proteins, so they will result into activation of complement proteins, attraction of phagocytes. So there is attraction of phagocytes because they can also bind to the phagocytes. On top of that, opsonization, increasing phagocyte efficiency. So you have opsonization that will result into increasing phagocytic efficiency or function of these phagocytes after opsonization of the antigen. Stimulation of inflammation, which is very obvious, and also prevention of bacterial and viral adhesion. So it's going to prevent bacterial or viral adhesion. Why? It's because the antibody will bind to the antigen binding site. It's the same binding site that the bacteria or the virus can use to bind to the normal cells for them to invade the cells. So if the antibodies are binding there, then they are preventing the bacterial or viral adhesion to other cells. Then responses to antigen exposure. So these responses to antigen exposure occurs in both cell-mediated and antibody-mediated immunity. The first exposure to result into production of initial primary response, then the next exposure, it will result into, or it will trigger the secondary response, which is more extensive and prolonged because of the memory cells that are already primed. So you have these memory cells that are already primed, So once you have the second response, it's very extensive and prolonged because you have a pocket of B cells, you have a pocket of memory B cells, you have a pocket of memory T cells. So the B cells will be activated into plasma cells, then they will respond by producing antibodies to that particular infection if you have a second infection. So this is just a summary of the function of antigen antibody complexes in a diagram. So you have viral inhibition. Why? It's because you can have a virus here that has got a binding sites. So the antibodies are specific to these antigens. They'll come and bind to the antigens, hence inhibiting this virus to invade other cells or to infect other cells. Antibodies react with molecules at the viral surface and prevent the viral attachment to the cells. Ready to say? Then the other function, neutralization. So this could be a toxin. A toxin has got a binding site. So before it injures any other cell, the antitoxin or antibodies can come and bind to this toxin. Then they're going to neutralize the toxin because the toxin now can be phagocytosed and it can't damage the cells. So these antitoxins, most of them are antibodies. So you have antibodies that can be harvested that are specific for a particular toxin. It could be a toxin that is coming from a reptile or maybe a snake or whatever it is. So you can administer these antitoxins in form of antibodies that can go and accumulate on that toxin and then they'll be neutralized by so doing. So antibodies called